Thanks for coming back. I think we had an excellent morning session with some great presentations and excellent questions and answers. And now we're going to move into the area that I think is really critical to all of this, is the plant itself and the importance of the plant. We're going to have to dig into that biology of the plant, both on the genomics side and the metabolomics side, as well as seeing how perhaps some of this information can perhaps move up and help those people in need in that field. So, just as a reminder, this is session 2. Is on the cannabis plant genomics and the agriculture for those who just joined us. We're here for the first session. We have 5 speakers today. They're going to be, again, that's the same format, making presentations between 5 and 7 minutes. And we ask that you hold your questions until the end, at which time we'll have an open opportunity to ask questions and make comments. To just presenting the panel today, on my immediate left is Dr. Philippe Henry. He's the Chief Terpenoid Officer of Vestal. To his left is Dr. Nolan Kane, Assistant Professor in the Department of Ecology and Evolutionary Biology at the University of Colorado Boulder. Greg Bodie, Director of Breeding and Genetics at Aurora Cannabis. And to his left is Dr. Sean Miles, Associate Professor, Department of Plant, Food, and Environmental Sciences at Dalhousie University. And on his left is Dr. Susan Murch, Professor, Department of Chemistry at UC. So, Philippe, if perhaps you can start us off with your presentation.
Absolutely. Thank you so much, David, and thanks to the organizers, UNC, for inviting us to come here today and chat about our favorite thing, which is the cannabis plant and the using genomics. Using targeted genomic markers to investigate the key traits in the cannabis plant, as well as genome regions underlying the therapeutic potential of the plant. I did stick to 5 slides, so they're quite populated. This is just a standard slide I like to start with when we talk about the cannabis plant, the diversity, the gene pools, where it originated, and essentially how humanity has interacted with the plant to shape the diversity that we currently observe. The map of the world shows here, essentially in small letter, three-letter acronyms, the different gene pools that are hypothesized, the hypothetical origins of drug-type cannabis, or the current cannabis that we have. People talk a lot about things like indica, sativa, ruderalis. In this case here, I talk more generally in terms of a large divergence in between European hemp used for fiber and the drug-type varieties. What you see with boxes are kind of the origin points, and at the bottom here you see a star, which points to the origin of, essentially based on paleological data of pollen, the putative origin of the cannabis genus in the Tibetan Plateau sometime about 28 million years ago diverged from the ancestral hop cannabis plant. So really the point of origin being the Tibetan Plateau, so the broadleaf drug point that you see up here as a purple would be what people would refer to as indica, and I have one shown here, kind of a style plant. And then we have the sativas, which are essentially thought to originate more from the tropicals, the narrow-leaf drug-type varieties, kind of like Sativa there. And then there's also Chinese hemp that originates here, BLH, so the broadleaf hemp varieties, so non-psychoactive varieties. And when we start looking into that point of origin, we see that there's tremendous diversity at these locations. And also what we do notice is the fact that this plant has traveled alongside humanity, and there's been a very strong divergent selection from the farmers for either fiber type plants or narcotic plants with very divergent traits. So either the drug type varieties, obviously cannabinoid expression, resin production, things like long fibers, kind of totally different structure of the hemp plants in terms of the architecture of the plant. And that existed naturally in a natural population, and we selected for different traits in these different groups. And again, what we see today is essentially a big mashup of these different gene pools and a lot of hybrid gene pools where it can become difficult to tease apart which trait came from where. But luckily, a lot of this has now had the tools of genomics applied to it, and we're able to now start teasing apart differences between the gene pools, particularly traits of interest for commercial reasons. One of the things that our team has looked at, and this has been done in collaboration with the lab in Las Vegas, Nevada, DigiPath Labs, was to focus uniquely on the drug type cannabis and essentially look at two things: the genotype, which was outsourced to a Boston company from the district Genomics that essentially hosts an open source cannabis genetic database called Cannapedia. And in addition to getting whole or genotyping by sequencing on a large number of accessions, we also got chemotype data for these accessions. So here's essentially a PCA that shows you three major groups in the drug type cannabis in terms of the expression of terpenes. So they're all either THC dominant, but you're also able to have, because the region of the genome that dictates the expression of THC or CBD is very localized, and that has recently been shown to be found on chromosome 6 of the cannabis genome map. You are able to actually have very divergent expression of cannabinoids, but still a very constant terpene expression. And these three different groups are illustrated here with different colors, and they're mostly dominated by a group that has beta-myrcene and beta-greatheline, which is also the most common group here, which is shown in red. And the group which is the most divergent, the green group, which is the terpinolene dominant group. And then the other group being more of a limonene, caryophyllene, pinene group. So based on a small number of chemicals, we're able to already get an initial signal from the upper level signal of how these drug type cannabis gene pools segregate. And essentially what we did, this is the data set that has about 10,000 SNPs. We were able to actually focus in and find which SNPs were the most informative using discriminative analysis of the principal components that essentially gives us now a set of 18 SNPs. It gives you the same signal as the genome-wide association. And what we want to use this for is a predictive tool to be able to essentially predict the expression of terpene expression in the next generation of plants. And this is something that has been tested and validated here in a larger data set where you see divergence using 18 to 20 markers where we can tease apart hemp and all the three different drug type cannabis varieties. And this can also be typed here. This is an example of a particular mutant that you can essentially type SNPs in a portable fashion in the lab, in the field, and essentially be able to get an individual barcode and predict terpene expression in the next generation. Here's my last slide, which I'll just leave it at. The same tool is applied to the human genome, where we're actually able to find different responses to people. So this ties into what initially was shown, and what I will quickly say as I'm out of time is that looking at it from a genome-wide panel, we were able to isolate all the variants that are found in the endocannabinoid system, and those are not predictive of people's responses to cannabis. And when we actually let the algorithm tell us which regions of the genome are associated with people's responses to cannabis, we found a totally different set of markers, including one marker found in the MAIA16 gene, which has been found in other studies to be associated with schizophrenia. And so that particular marker was always homozygous in people that have a very negative response to cannabis. So that's all I got to say for the human side, but I'd love to chat more in the question section about that.
long-standing puzzles about the effects of cannabis. And the basic things I wanted to think about are the two basic things I wanted to think about are long-standing conflicts between general knowledge about cannabis and what's been able to be validated extensively in the medical literature, and a related puzzle that is basically the lack of adoption of the existing cannabis-derived pharmaceuticals, despite it being such a widely sought-after plant for the medical and the recreational side. And some of this is already well known, but I still think it's worth exploring a bit more. And being an American, I have to confess to being an American, and harsh audience. I have to admit that the American view of cannabis is heavily shaped by the same agency, the Drug Enforcement Agency, that is charged with preventing the use of any illicit drugs. So for 50 years, actually more than 50 years now, the sole source of cannabis for the U.S. scientific studies has been the University of Mississippi grows it for the National Institute for Drug Use, which is the sole source of federally approved cannabis for medical studies. And I think that is basically intimately related to much of the literature not showing strong effects for many widely reported effects of cannabis. And to just give you a little bit of a taste of what I'm talking about there, so just last year there was a pretty thorough review by the National Academies of Sciences, Engineering, and Medicine, so very high-profile scientific and medical organization, did a pretty thorough review of literature and all kinds of findings, but the main finding was actually the lack of support for so many of the widely accepted effects of cannabis that I found absolutely astounding. And the two things that I would highlight as being the most surprising to me of their findings are that they were not able to support. They found basically insufficient evidence for effects on appetite, which I found absolutely astounding because that's, I mean, haven't they ever heard of the munchies? I mean, come on. And I found it absolutely astounding that they did not have good scientific support in their review of that. And then they also weren't able to find good support for effects on intractable epilepsy and many other widely accepted uses of cannabis. And intractable epilepsy is in many respects very widely accepted to the extent that, for instance, in the state of Utah, it's the only legal way to possess any sort of cannabis is if you have very particular types of intractable epilepsy. And so if they're not able to find support for that, and yet quite conservative lawmakers are basically disagreeing with that, I find that to be surprising. All right, so just to cut to the chase a little bit, I'll point out that, so this is just a subset of the well over 1,000 lines that we've looked at genomically, so just a few hundred of them, but I think it's pretty representative of our findings. And in cannabis, there's multiple different distinct lineages, as you've heard from others. And our analyses show pretty strong evidence that there are quite well-supported lineages, broadleaf drug types, narrowleaf drug types, and hemp types. There are also multiple distinct lineages within those. Basically, all of those have distinct lineages within those, but there's at least 3 broader lineages, and I think if we had better sampling, we'd also find ruderalis as its own separate lineage. But also, almost all of the plants they looked at are hybrids as well, so it's even more complex than that. And support for strong differences between these lineages in at least 9 cannabinoids and multiple different terpenes. So these are genetically based differences, but they also are associated with some important trait differences. So I would say that there's a tremendous amount of diversity within the plant, as everyone seems to be pointing out these days. And where does NIDA material come from? The sole source is basically, it would be one of these lines here. Basically, all of the material that we've looked at so far are closely related, low quality hybrids between hemp and sativa types. Basically, hemp by Mexican sativa varieties. So very low diversity, even though it's a hybrid as well. So basically, if I want to summarize the chemical differences between it, I'll just briefly state that they have on average about a quarter the level of THC for all the high THC varieties, much lower CBD than you get in any of the U.S. state legal markets. No measurable amounts of any minor cannabinoids. Also, no measurable amounts of terpenes reported in the material that they give out to researchers. However, they do have a lot of something, and that is CBN, which is they have about 20 times the level of CBN as anything you get in state markets, which that's very, very poor storage conditions, because that's the predominant breakdown product of most cannabinoids. So that's the material that virtually all of the U.S.-based research is based on. And so, to me, that suggests major caveats. Here's what they look like. And this is from material provided to researchers. This is a photograph of her material after years of work trying to get the approval and the funding to do this research. She got this stuff. And she's trying to do research that's supposed to be relevant to this stuff. Completely astounding. All right, so just to briefly summarize, there's at least 3 major lineages of cannabis, including hemp and multiple drug types. They differ in all kinds of chemicals that are known to be important for health, and the stuff that a lot of the research in the U.S. is done on is not relevant, essentially, to what you guys are doing here in Canada now that it's legal, or with any of the state markets in the U.S. Because of these challenges between the mismatch between what is studied in a lot of the literature and what's actually used by most of the public, we're only beginning to understand the effects of the most widely used, one of the most widely used medicinal plant. And I would urge extreme caution any time you read one of these review papers that says cannabis does this or cannabis does not do this. Keep in mind that a lot of those statements are heavily based on this research that is on a product that is so different than what most people are using that it becomes almost irrelevant. So, unfortunately, you have to take most of the medical literature that relies on anything coming out of the U.S. with a huge grain of salt. So just pointing out, and I think that explains why they're not able to find a lot of effects that are well documented anecdotally by many physicians and people on the private side. So, yep.
I can see why people are having trouble with this. The forward arrow is completely worn off. So I'm always excited to talk about genomics and crop improvement, and I think cannabis is kind of in a unique and pretty special position right now, because I really think that a major chapter in the crop improvement of cannabis is going to be written over the next 5 to 10 years. And unlike most of our other crops, when that chapter was written decades ago or 100 years ago, for this case, genomics is going to be a fundamental part of that chapter. So it might be one of the first crops to have that be part of it. So really exciting. So my title was something to do with cannabis genetic diversity, but we have just talk a little bit more about cannabis cultivation and about Aurora, of course. So I'm the director of Green and Genetics at Aurora. So for those of you who may not be familiar, Aurora is a Canadian company that's also a global player in cannabis. So we have production across the country, and that's a mix of both indoor, outdoor, industrial hemp. These glass house, modern glass houses, which I'll show some pictures on in a second. And we're also international, so we're across the world in a number of different countries. And that's both producing industrial hemps, processing, clinical trials, medical production of medical cannabis. So there's all those pieces there, and all of those things rely on having good production, and that relies on having good genetics. And that's what me and my team are working on, is producing better genetics for Aurora cannabis. So historically, at least in North America, cannabis has been produced in kind of two formats, at least it's my understanding. In Canada, we had a lot of indoor production. So, back in the day, that was basements, warehouses, and super high labor, lots of, all 100% artificial lights and sort of makeshift operations. And the other sort of scheme is, at more southern latitudes, is big outdoor photoperiod sensitive plants that flower late in the fall, and are these great big Christmas trees. This is a picture of one of Aurora's facilities, and the type of high-tech greenhouse that we're building both in Canada and elsewhere. So this is called Aurora Sky. They all have kind of names of the sort of elemental nature, I guess. It's about 1 million square feet. So people who, just a quick conversion you can do is it'd be about half the number of plants as there is square feet. So it's about 500,000 plants in this greenhouse at any given time. And it's a hybrid greenhouse structure. So it's really a grow chamber with a glass roof. So the entire building is under positive pressure. All of the air intake is completely controlled. There's no open ventilation. Of course, there's full supplemental lighting. You can see blackout curtains moving along there. And the other thing you can see is especially in the top right corner, is the plant density is very, very high. And there's actually not a lot of room even for people to walk around and look at plants, because it's based on an automation model. And you can see the bottom there. Basically, a robot going out and picking up the whole table of plants, where in this case it's moving from high density veg, so you can see the plants are pretty close together, and it moves through a room where another robot actually re-spaces them out to a lower density so that they can go into the flowering room. Of course, there still is a lot of labor. You can see all the hand trimming going on here. So there's still a bit. And of course, the other labor-intensive component is cloning. This is a completely novel environment for cannabis to be grown in, so there's a lot of challenges that go along with getting the cultivation this right. Of course, genetics is going to be a big component. I've sort of irradiated this to have useful information, wearing my industry hat. But just to say that, like a lot of other speakers here today, we're collecting a lot of genomic data and a ton of phenotypic data in a more rigorous way than previously been possible, because we have access to licensed grow space where we can actually grow trials of plants. And this genomic data is going to be the sort of foundational piece to our breeding program. And this is where we're going to be doing most of our breeding. We're going to be using some of the other Aurora sites. I'm super excited about this. This is just a few weeks ago, and just a few months ago, I was still sharing sort of 3D renderings of the building. So I'm really happy this is an actual picture now. So it's a 10,000 square foot research building. So molecular lab, phenotyping lab, office space, and then 22,000 square feet of greenhouse, and this facility is only for breeding and genetics. So it's 100% R&D facility. So, thank you.
simple message, and it's right there. So, many of you are familiar with how cannabis is labeled. When you go to purchase, when consumers go to purchase cannabis, it's very common for them to be labeled with indica or sativa, or then some kind of proportion thereof, 75% of one, 25% of the other, and so on. And what I'm here to tell you is that according to the data that we have collected and analyzed in our small genomics lab in rural Nova Scotia, we have determined that the labeling is not an accurate representation of what is inside the package. It does not give you any meaningful information about the genetic identity of those plants. So let me just review this a little bit. The first study that we published in PLOS One, together with a grad student, Jay Soller, led this. This was in collaboration with Anandia Labs, which is here, a research company here in Vancouver. And Jay is now a bioinformatician with Anandia. And in this study, so what we did is we looked at 85 different strains, if you will, or varieties of cannabis, and we look at their genomes. So we genotype markers for genomes. You want to get a good representation of the genome. We've done so with 14,000 genetic markers across 85 different samples that were obtained, either from licensed producers. This is in the early days. This is even very, very early days. And then, or from seed catalogs and so on. And each of these samples they provide you with, okay, a label that says it's 100% sativa or 100% indica or something in between. So to investigate the degree to which those labels actually correspond to anything meaningful that we collect from the genetic data, here's one way of doing this. And we can view how samples are related to each other in 2 dimensions using this technique called principal components analysis. So in general, all you need to know here is that each of the dots on this plot represents one of the strains that we analyzed. There are 85 dots on this plot. And they're colored by how they were labeled when they came to us. So the ones that are dark orange were labeled as 100% sativa. The ones that were labeled dark blue came to us and were labeled as 100% indica. And then there are varying degrees in between, you can see, according to the shade. Now, the distance between the dots here in this plot essentially gives you a rough representation of their genetic distance from each other. So indeed, you do see that, to the right on this plot, you find exclusively 100% sativa varieties, and on the left-hand side of the plot, you do see most of the indica. And so there is a rough concordance between the label and what the genetics are telling us, okay, what the genetic identity of the plant. But the concordance is very rough. It's not a very good representation. You can see here in the top of the plot, in the top left, for instance, there are 2 strains there. One is labeled as 100% indica, the other one is labeled 100% sativa, yet they are more closely related to each other than they are to any other sample in the data set. I'm glad you're laughing, because in any other agricultural crop, you can't do that. So you can't stick Macintosh apples in the bin at the grocery store and charge $3.99 and call them Honeycrisp, right? And you can't put Pinot Noir in the bottle and call it Cabernet Sauvignon. It's a similar sort of thing. So we undertook another study currently that is not yet published, but should come out this year or next, with Bedrocan, looking at strains from Dutch coffee shops. So again, we have about 25,000 markers in this case, so a good representation of the genome, about 150 different strains, and these were labeled as either indica or sativa, and this is work that was led by Michael McElroy, who's a postdoc in my lab, and he's now a winter wheat breeder in Quebec. So here's another way of representing these data. So we can take from these 150 strains, we can say, when we see, we compare every single pair of strains. And when we're comparing one that is labeled as 100% sativa to another one as 100% sativa that's labeled as 100% sativa, we look at their genetic distance. We look at all pairs of all samples that are labeled as 100% sativa, and we get that red line. That's their genetic distance across those samples. So you can see it's sort of centered on about 0.3. And then we look at all pairs that are labeled as 100% indica and 100% indica. And we go and we draw that distribution, and you see the yellow one. Then we take all the samples, all pairs of samples that are labeled opposite, 100% indica and 100% sativa. Now you would expect that distribution to have genetic distances that are far larger, right? But you can see that's the gray line, and it looks no different than the other two lines. So now, the basic message is, according to the data that we've collected in our lab, in any case, from 2 different samples, one from Canada and one from the Netherlands, that the labeling practices of using the words indica and sativa, and by no means am I suggesting that indica and sativa don't somehow botanically exist. All I'm saying is that what's currently on the labels is not a meaningful representation of the genetic identity of these plants. Thank you.
Hi. Hello. Okay, so I'm going to start with a couple of really quick questions, just because I feel like we've been sitting very patiently for a while. So, all the people in the audience, can anybody tell me, and don't push your buttons, because I have a vision that if we all push our buttons at once, the whole building's going to collapse so I can't start that. Okay, so, just shout out a number. How many unknown chemicals did you eat today? 72. Okay, anybody know? How many chemicals are there in an average leaf? How many chemicals are there in cannabis? So my research program looks at the chemistry of plants and how plant chemistry affects human health. I'll tell you that in an average leaf there are about 30,000 distinct chemicals. We can identify somewhere around 1,200. We estimate cannabis to be somewhere between 25 and 30,000. It could be more. And you eat about 200,000 unknown plant chemicals each and every day, which is the standard pitch I use, which is we really need to know a lot more about plants if we're going to know anything at all about foods. So my work in cannabis and the work of my students on cannabis is really about how do we understand that chemistry and then how do we make decent food. And I don't really think we have any idea. I really think no one knows. So this opinion piece in the Hill Times is, they count down to the rocket launch of how we're going to have edibles in the marketplace. But there's so much we don't know that we really don't know. So this is what we do know about cannabis. Last time I counted, we can come up with the standard set of about 1,000 primary metabolites, that's all your amino acids, sugars, and the things we need to grow. And then about 560 additional. So those 560 break down into things like flavonoids and isoprenes. When was the last time anybody said to you something about flavonoids? That's why they're purple. Terpenes, cannabinoids, always mess out when I was talking about CBDs. THC is the least interesting of all these molecules. By far the least interesting. Unidentified flavor and odor compounds. All kinds of things that have effects on human health. So my PhD student, Elizabeth Murch, and my collaborator, Paul Brown, and I worked together, and we worked with 5 different licensed producers. This is a study that analyzed 33 commercial strains. All of them have different names. What do those names mean? Now, I'm real famous because I like to poke a little bit at students, and they'll understand my sense of humor. So I ask questions like, what's a species? Are there really different species? I also think the genomics part's the easy part, because there's really only 4 chemicals in the genome, and you can boil them. So that's not at all hard. These ones are not stable. We got to do fancy things to get that. So we analyzed 32 CBDs, 29 monoterpenes, and 38 sesquiterpenes across the study, and that gave us a pretty rich data set. We clustered them and looked at, were they really different? 24 of the strains, there's more variability within the plant than there is between plants. So they're really not all that different. But then in that field of forces, there's a few zebras. And the few zebras are what are really interesting. So the known stuff is all there in the pathway. This is published information, by the way, if anyone would like the reprints, I'm happy to send them. But what was new? And I apologize for the title, but apparently moved. So what was really interesting was we found some unknown structures that had previously unidentified and identified some of those, and those were associated with specific strains that people are reporting specific uses for. So what I think is interesting there is we have the capacity to link that human use data, which is really anecdotal, or gray market data, and make it correlated to some of the chemical data. And so that's one of the hopefully future applications of the work. Now, we just put out a paper last week, and I'm a little bit cheeky, I told you that. But what I asked my student was, is it possible that in all that gray market breeding program, what they really selected for was how the plants smell? Is there some odor there that the breeders thought was linked to THC? And if you listen to the stories that are told, this is often something repeated. So is that true? Well, it turns out the high CBD varieties are also high in non-specific terpenes, and the high CBD ones are high. Let me try that again. The high THC ones had 9 terpenes correlated, and the high CBD ones had 7. And they were different ones. So the CBD was slightly more floral, slightly more citrus, a bit less musky. Did people do that, or did plants do that? It's hubris to think that the plants did it for us. So I always like to end everything I say by thanking the people who really did all the hard work, and the work I just presented was actually done by Dr. Paula Brown and Dr. Beth Murch, who are a fierce group of chemists and really interested in what's in the chemistry. So thank